Nowadays we use crop improvement methods by means of plant breeding. So what are the various steps that are required for bringing or developing these new varieties? They are as follows. First of all, it is important that we need to collect the germplasm. What is germplasm? Germplasm is the sum total of all the alleles of the genes present in a crop and its related species. So we need to collect the germplasm in order to have a successful breeding program. So plant breeding's first step involves, that is, the collection of germplasm. Means, what is germplasm? Remember, it's the sum total of all the alleles of the genes present in a crop and its related species. So when we collect the germplasm, it means we are moving towards a successful plant breeding program. Next, we have evaluation and selection of parents. Now this is very important. The parents that you are going to select, they should have the desired traits that you require, right? So for example, if you require longer grain rice, then you need to select parents that actually have the desired trait of having longer grain rice. So it's very important that you select parents on the basis of the desired traits that you want, right? So selection of parents is very important. The type of parents you select for this plant breeding programs, the result will be in that sense. So you need to remember that the parents you select should have the desired traits that you want in the progeny. Next, we have cross hybridization among selected parents. Now what is hybridization? Hybridization means crossing of two or more types of plants. Now they can be two plants also, two or more types of plants also, for bringing their traits together in the progeny. So we want the desired traits in the progeny, so we are going to select the plants, we are going to hybridize them, means we are going to cross them with each other. They can be two plants, they can be two or more types of plants. We are going to bring their traits together in the progeny. This is known as hybridization. Now for hybridization, we need to understand what do we mean by lines, or you can say pure lines. Now pure lines or lines means group of individuals that have similar genotype. So they are pure line. And in case of hybridization, we also call pure lines as parents. What do we call them as? We call them as parents. So as I told you earlier, the second step, that is the evaluation and selection of parents. So you select parents on the basis of the desired traits that you want to achieve in the progeny. The same way out, we sometimes use pure line plants, means we use the group of individuals that have similar genotype, right? These are pure line plants that we call as parents, and then in the progeny, after this cross hybridization, we achieve the desired traits that we want for this plant breeding program. Now what is the procedure of hybridization? Let's discuss the procedure. First of all, selection of parents with desired character. So as a whole, everything revolves around the selection of the parents. So the desired character should be there in the parents. As I told you, for example, you want longer grain rice. So you will select the parents based on what you desire in the progeny, right? So selection of parents with desired characters is very important. This is the first step of hybridization, remember this. Then secondly, it is selfing. Now what is selfing? Selfing means we are actually doing what? We have parents and we are crossing the parents, means we are doing selfing. For example, we have crossed the parents, and this is, for example, say capital T and small t, and we have this progeny. Now when we cross this with the same, this means we are doing selfing. So crossing with each other means we are doing what? We are doing selfing. So selfing is very important. After selfing, we need to have emasculation. What do we mean by emasculation? Emasculation means removal of anthers from the flowers of the plant before they become mature. So what is the main aim of the removal of anthers? Because as you know that anthers contain the pollen grains, and the pollen grains, if they are allowed to mature and they are going to fall on the stigma of the same flower, then we will not be able to achieve the desired result. So what do we want is, we are going to remove the anthers from the flower so that the pollen grains are not mixed, right? So that we can actually control self-pollination. I hope you know what is self-pollination. Self-pollination means when the pollen grains from the same flower are going to enter on the stigma of the same flower, that is self-pollination. But by doing emasculation means by removing the anthers from the flowers, we are going to assure that there is no self-pollination in the flower, right? That there is no self-pollination in the flower. We are going to prevent this by doing emasculation means by removing the anthers from the flowers before they mature. Then we have bagging. Now what is bagging? When we have removed the anthers, we are going to cover that flower by means of some paper, by means of some polythene, so that no other foreign pollen grain can even enter in it. Because we want to get the desired results, and for that we are going to provide those pollen grains that have the desired traits, that have desired characteristics, right? So we don't want any foreign pollen grain to enter that flower, so we are going to bag it, means we are going to cover that flower from which we have already removed the anthers so that no foreign pollen grain can enter. After this, we have tagging. Now whatever procedure you are following, it's very important that you do it by tagging. How tagging? Just mention the date, mention the time, so that you can have a record of everything what you are doing. So tagging is also one of the important part of the plant, of this hybridization procedure. Then we have artificial pollination, that we also call as crossing. Now why we are calling it as artificial pollination? As I told you, because we have done emasculation, means we have removed the anthers from the flower. So we want that we are going to provide those pollen grains that have the desired characteristics. So this is known as artificial pollination, right? So as the name suggests, we are going to supply the pollen grains on the stigma of the flower, right? The desired traits are going to be there in the progeny because of this artificial pollination, which we also call as crossing. Now artificial pollination is done by means of taking a brush and we supply pollen grains on the stigma. We dust pollen grains on the stigma of the flower so that after that fertilization can occur and we can have the progeny with the desired results. This is how artificial pollination is. This is how we call it as crossing. So these are the steps for the procedure of hybridization. Means first of all, just select the desired parents with the desired characteristics, then select selfing, then emasculation, bagging, tagging, and then we do artificial pollination that we call as crossing.